Hi, Miss Nikki here. Welcome to the urinary system. This part one is very anatomy heavy. I wanted you to see the difference between real anatomy and then kind of this textbook version, graphic design anatomy. So things are not quite as easy to differentiate. We can still see the kidneys and the adrenal gland, and you can see the ureters or ureters, but realize that, looks a lot different in real life than it does in these models or in the textbook. We know the urinary system consists of kidneys and ureters or ureters, the bladder, and the urethra. So the kidneys are actually going to filter the blood. The ureters or ureters are going to transport the urine to the bladder. The bladder is a muscular sac that's just going to fill and store urine. And then the urethra is what we're going to use to eliminate the urine from the body. One of the things that we'll think about in the kidneys as we walk through this chapter is that we do have the ability to filter our blood, but then we can reabsorb, do reabsorption or secretion as that filtrate is moving through the kidneys. They wanted you to look at the posterior view of the kidneys and pay attention to the fact that we have our floating ribs there to protect our kidneys. They wanted you to see that the right kidney is a little bit lower than the left kidney. It has to do with the fact that the liver would be on the anterior side, so it kind of downshifts a little bit. Wanted to show you the ureters or ureters and how they come through and deliver urine to the bladder. Looking at the functions of the kidneys, that they want you to realize that the kidneys do a lot of processing. So the blood is going to enter the kidneys, it's going to become filtrate, and then we're going to do that reabsorption or secretion. We're going to get rid of metabolic waste, so any byproduct, urea, uric acid from the breakdown of proteins and nucleic acids. We're going to be able to regulate the ions. So if we need more of them, we're going to reabsorb them. If we have too many, we can excrete them. The acid-base balance regulation. So we're going to learn when we do acid-base problems that the urinary system can compensate for the respiratory system. So we had this equation. So carbon dioxide with water forms carbonic acid, which then gets broken down into bicarbonate and hydrogen ions. So you can excrete these bicarbonate ions or hydrogen ions in the urinary system, and it can compensate if you have too much CO2 or not enough CO2. So remember, we talked about respiratory acidosis and respiratory alkalosis before. After this chapter, once we get through urinary, we'll talk about fluid and electrolytes, and we'll talk about how one system can compensate for the other strictly because of this equation that we learned in respiratory. We're also going to see the regulation of blood pressure. So hopefully you are going back and thinking about things like ADH, aldosterone, renin, atrial natriuretic peptide. And then we're going to see some biological molecules that we've consumed, like drugs or hormones that we've made. We might be able to get rid of those, right? We don't want them to stay around forever. They're going to break down, and some of the products or the waste from the breakdown of those hormones can be excreted in your urine as well. Other functions of the kidneys, we have the formation of calcitriol. This increases calcium absorption in the small intestines. If you remember, this works in conjunction with parathyroid hormone if your calcium levels are too low. It also creates and releases EPO, and remember that increases erythrocyte or red blood cell production, and it can also engage in gluconeogenesis. We learned about this when we talked about cortisol in the adrenal gland. Due to prolonged fasting or starvation, the kidneys can also produce glucose from non-carbohydrate sources like proteins and fats. Remember, glucose is required specifically for cardiac muscle and then brain. They really like, both of these really like glucose only. Anatomy of the kidney. So the kidneys, there's two of them. They're symmetrical. They're bean shaped. You're going to see this term helium again. So we saw this in the lungs. It was kind of the entryway point. You're also going to see the adrenal glands, the hats that sit on top of the kidneys. Very detailed kidney location here. It's on the posterior abdominal wall. It's lateral to the vertebrae. Left kidney is between the 12th thoracic vertebrae and the 3rd lumbar. The right kidney is slightly lower than the left. They're partially protected by the rib cage. They are posterior to the parietal peritoneum. And if you remember, the parietal peritoneum is that serous membrane that reduces friction, right? And it's on the backside. So we call the kidneys, their position, retroperitoneal. So the peritoneal membrane would be here, and they are on the outside of it, behind it. So remember, you've got parietal peritoneum, then you're going to have the cavity, peritoneal cavity, and then you're going to have your visceral peritoneum. When we look at the tissues that are supporting the kidneys and keeping them in their place, there's a fibrous capsule, and it has that dense, irregular connective tissue. It helps maintain the shape, protects it from trauma, stops pathogens from entering. Then you're going to have a layer of fat. This is going to cushion and support the kidney. Then you're going to have another layer of dense, irregular connective tissue, and this helps anchor the kidneys to surrounding structures, and then another layer of fat, and this is also going to cushion and support. So basically, you're going to have a fibrous layer, then you're going to have a fat layer, then you're going to have a fibrous layer, and then you're going to have a fat layer. So several layers of connective tissue for anchoring, and then several layers of fat that are going to help cushion and support the kidneys. And this image here is just showing you the fat surrounding each of the kidneys and the renal fascia and the fibrous capsule. In this clinical view, we're looking at what happens if the kidney moves inferiorly. So if something's wrong with the connective tissue, if you happen to lose a lot of adipose tissue, you're going to see this renal ptosis. So the P is silent. It causes the kidney to move down inferiorly. It can cause the ureter, remember ureter leading to the bladder, that can kink. It can block urine flow. You can actually have urine backing up, and that would cause a swelling of the kidney, which can lead to renal failure. And you can see this root word. We're going to talk about the functional units in the kidneys, and it's called a nephron. So this hydronephrosis would be the swelling of the kidney due to the backup of urine. When we look at the sections of the kidney, there's always going to be an outer. So the outer portion is the cortex, and then we're going to have the middle portion, which is the medulla. And we've seen these terms before. We're going to have renal columns and renal pyramids. So inside the medulla, you're going to have this pyramid-shaped structure. And between each pyramid, that would be a column. These would be the pyramids. And then at the end, you're going to see this rounded structure, and that's the renal papilla. We have additional structures, the renal sinus. Think of this more, I guess, drainage. Yeah, the word's right there. This is a drainage area composed of minor calyces, major calyces, and the renal pelvis. It's a little bit easier to see on an actual image. So here we have the cortex is the outer ring. All of the internal would be the medulla. You can see the pyramids here. There's several of them. And then in between each pyramid is the column. You can see the renal papilla right here, the rounded portion of the renal pyramid. And all of it dumps into all of this, which is called the renal sinus. Then we can look at the fact that we have this minor calyx and this minor calyx, and they merge into a major. And then several majors merge into the renal pelvis, and eventually that drains to the ureter or ureter. So kind of the flow here is if I had renal papilla here, is going to drain into a minor calyx, which is going to drain into a major calyx, which is going to drain into the renal pelvis, which is going to drain into the ureter. That's going to lead to the bladder, and that's going to lead to the urethra. Each kidney is innervated by both divisions of the autonomic nervous system. Sympathetic nerves, hopefully we still remember fight or flight. This is going to decrease urine production. We're really kind of still unsure about what the vagus nerve tells via parasympathetic pathway, so we don't really know. So the nephron is the functional unit of the kidney. The nephron consists of several different sections of tubing. So we're going to name all the parts of the tube, but the entire structure is called a nephron. So this entire structure from here on over, so not including these two sections, all of this is a nephron. The nephron includes the renal corpuscle, which there are two parts. We have the glomerulus, which is the first part, and we have the glomerular capsule, which is the second part. So this is the glomerulus, and we can see the capsule around it. Then we have this tube section called the proximal convoluted tubule. It is directly off of the glomerular capsule. Then we see this loop of Henle, or nephron loop as it's called now. And then we have this distal convoluted tubule, and they all lead to the collecting duct. So the renal corpuscle has two structures, the glomerulus and the glomerular capsule. The glomerulus is a tangle of capillaries. Blood enters via the afferent arteriole and exits the efferent. The glomerular capsule has a permeable layer and an impermeable layer and a space between that receives the filtrate. So the blood is going to enter into the glomerulus, and filtrate is what comes through. So small items, and we'll see an exact list in a second, small items are going to filter through the glomerulus, and they're going to get caught in the glomerular capsule. So coming from the blood through the glomerulus, which remember is capillaries, small items are going to filter through the capillary glomerulus. They're going to collect in the glomerular capsule, and we now call it filtrate. Tube system, there's the proximal convoluted tubule, the nephron loop, and the distal convoluted tubule. The proximal is the first region. It comes right off of the renal corpuscle. It's made up of simple cuboidal epithelium. Hopefully we can go back to our histology. So one layer, cube-shaped cells. If it's epithelium and it's one layer, it's probably going to have something to do with absorption and secretion. You're going to see microvilli. They're going to increase the surface area, lots of surfaces to absorb or secrete something, and we're going to see a lot of reabsorption in this first proximal convoluted tubule. Nephron loop. So it has a descending section of the loop, and then it has an ascending section of the loop. PCT would be before, DCT would be after. We're going to see the distal convoluted tubule after the nephron loop. It extends to the collecting tubule, which several nephrons are going to eventually dump from collecting tubule into collecting duct. This structure is showing more of what it looks like actually in the kidneys rather than that elongated structure that we talked about before. So here you can see the afferent arteriole is delivering blood to the renal corpuscle. We can see the blood is traveling through the glomerulus, which is the capillary, and what's coming out into the proximal convoluted tubule we call filtrate. It's going to be all of the small items. Any large items are going to get pushed out of the glomerulus into the afferent arteriole. So once the filtrate hits the proximal convoluted tubule, it's going to come to the loop of Henle or the nephron loop. Then it does distal, and you can see distal convoluted tubule dumps into this, which is the collecting duct. And I like how they're showing you here that there would be more than just one nephron. There'd be multiple. There are millions of nephrons. Here's the same structure that we saw earlier. Renal corpuscle composed of the glomerulus, the glomerular capsule. Then we have the proximal convoluted tubule, we have the nephron loop, we have the distal convoluted tubule dumping into the collecting duct. So again, in the afferent, it's delivering blood. Where did this blood come from? It came from the renal artery. We'll see the blood flow in just a second. So renal artery goes through several other naming of the artery as we're moving in. The blood eventually gets to the afferent arteriole. All of the smaller items are going to move from the glomerulus through the pores into the glomerular capsule and eventually make their way to the proximal convoluted tubule. Anything that comes through is called filtrate. The larger items are going to get pushed into the efferent arteriole back into the bloodstream. There are two types of nephrons. So we have cortical nephrons, and that term cortex is in there. And this is about 85% of our nephrons. They have short nephron loops. They're barely penetrating into the medulla. The juxtamedullary nephrons have really long nephron loops, and they go deep into the medulla. And we're going to see that this area allows for the regulation of urine concentration. And this is coordinated by ADH. So go back and review ADH. If you remember, if ADH is on, that's going to cause the reabsorption of water. If we reabsorb water, you're going to have more concentrated urine. What happens if ADH is off? You're not going to reabsorb as much water, and you're going to have more dilute urine. So a nice comparison here, you can see the cortical nephrons. This is about 85%, juxtamedullary nephron about 15%. You can see the short nephron loop barely in the medulla, and then you can see the very long nephron loop going all the way into the medulla. I also want to point out these purple are capillaries. So remember we said we're going to reabsorb from the tubule back into the bloodstream. That happens at these capillaries. Nephrons are going to drain into collecting tubules, and then several nephrons via the collecting tubule will drain into larger collecting ducts. We're going to see that there are specialized cells in the collecting ducts. So in the collecting ducts, you have principal cells that are responsive to hormones, aldosterone and antidiuretic hormone. If you remember aldosterone, if it's on, you're going to have sodium reabsorption, which is also going to cause water reabsorption because they're co-transported, right? Antidiuretic is just water reabsorption. You're also going to have some cells that are going to help regulate urine and blood pH. So types A cells and B cells can individually reabsorb or regulate bicarbonate and hydrogen ions. So hopefully you're seeing the connection between the kidneys being able to excrete these ions back to that equation that we drew with carbon dioxide and water forming carbonic acid. Just to show you the histology, the renal cortex, renal medulla, what does it look like? They're showing you the actual microscope image of a renal corpuscle and showing you the tube structures. And then here in the renal medulla, they're showing you what do the sections of the nephron loops look like, what does a collecting duct look like, and that there are capillaries wrapped around these tube structures. So the juxtaglomerular apparatus helps regulate the blood filtrate formation and systemic blood pressure. A lot of people call it JGA. You're going to see cells called granular cells, and these are modified smooth muscle of the afferent arteriole. They're near the renal corpuscle, and when they're stimulated, they can release renin. So hopefully we're thinking granular cells can release renin. Renin goes through that angiotensin 1, angiotensin 2, which then triggers aldosterone release. And aldosterone release is going to increase sodium absorption, increase water. Would that increase blood volume? Would that also increase blood pressure? Yes. There are also a group of cells called the macula densa in this JGA. These are in the wall of the distal convoluted tubule, and they detect changes in sodium chloride concentration. They can signal the granular cells to release renin. So hopefully you can think about what happens if my sodium chloride levels are too high, what if they're too low. We know that these are held together by ionic bonds, that they can break apart in water, and that somehow we're measuring our sodium level. So why would these macula densa want to talk to granular cells to release renin just by monitoring sodium? If the sodium was too low, could we trigger the granular cells to release renin to make or have the release of aldosterone? We have to go through that angiotensin 1-2 system, but have aldosterone released so that aldosterone would increase sodium and therefore increase water as well. There's this extra area called juxtaglomerular mesangial cells. This is outside of the glomerulus. It's between the afferent and the efferent, and they think that it communicates with the other cells in the JGA apparatus, or JGA overall. They don't really understand the system too well. It's a group of cells between the macula densa and the granular cells. So let me do different colors here so we can kind of go through this. Here are those extra mesangial cells, and you can see they're in this pyramid shaped right between the efferent and afferent. And then we can come to look at the macula densa. So the macula densa are right here in the distal convoluted tubule. And then at last we have the granular cells, and the granular cells are right here in the afferent arteriole. So remember, macula densa can monitor sodium levels and trigger granular cells to release renin. Granular cells are also monitoring the blood pressure as the blood moves through the afferent arteriole. So then the granular cells, if they notice a disruption in blood pressure, they can also stimulate the release of renin and have increased water, increased sodium due to aldosterone. So in this elongated version, they're showing you the entire pathway, but over here, they're showing you that this is the distal convoluted tubule, and that right here you would have those macula densa cells. And then here in the afferent arteriole is where you'd have those granular cells. So one of the things to think about is location, and I kind of went over this with the last slide. The afferent arteriole, where is the blood coming from? It's coming from systemic, right? This is a renal artery, and that blood is flowing into the afferent arteriole here. So does it make sense that we have cells that can monitor blood pressure right here? So they can release renin if they need to, and they can increase blood pressure if the blood's not flowing in the kidneys fast enough. So this is kind of like a localized way to control blood flow. These macula densa are here at the DCT. So this part will loop back. Remember, this is the elongated version, not the one where it's all coiled up. So here in this distal convoluted tubule is where you have the macula densa. So I think of this as kind of my last chance to do any sort of water regulation. So in this part, in the PCT, I'm mostly doing absorption, and I'm being very general here. Nephron loop, this is going to have something to do with water reabsorption. This is going to be electrolytes, sodium, potassium, calcium. And then here in the distal convoluted tubule, I'm also doing absorption. I might be doing some secretion. I have too much, put it back into the tubule to go out as urine. But we also have these macula densa cells as kind of the last ditch effort to look at my sodium level and maybe do something about it. This is the collecting duct, and this also responds to ADH and aldosterone. So these two are going to have something to do with water reabsorption, and these two are more going to have to do with reabsorbing ions or excreting ions. So we'll now look at the arteries just so we can see kind of this pathway. The renal artery comes off of the abdominal aorta. The renal artery is going to differentiate into the afferent arteriole that leads to that renal corpuscle. And then we're going to see the efferent arteriole is what exits the glomerulus. We're going to see capillaries. If the capillaries are wrapped around the PCT and the DCT, peri around the tubes. Vasorecta means straight, right? These are going to be associated with the nephron loop. So all blood is moving through the capillary beds. It's filtered by the glomerular capillaries, passes through the peritubular capillaries, the vasorecta for gas exchange, nutrients, and waste, and then finally drains into the network of veins. The veins, I don't have these highlighted. We're going to look at the veins leading to the renal vein and then draining into the inferior vena cava. So this looks kind of complicated. Remember, we're not going to do all these. So I just want you to see renal artery, and I want to make sure we know this comes off of abdominal aorta. So we're going to see the blood from the renal artery, and these are the sections that it flows through. We're not going to have to name these. We know that eventually the blood is going to flow into the afferent arteriole. And then the blood is going to be filtered by the glomerulus, right? And the blood, any small particles are going to drop through the glomerulus capillaries into the capsule, right? So we've created filtrate. Those small items are going to move into the proximal convoluted tubule. Anything that's large gets ejected into the efferent arteriole. So then we have our efferent arteriole right here, where the blood, any large like red blood cells, white blood cells are being pushed out. Then we're going to see this continues and becomes peritubular capillaries and vasorecta capillaries. So if I'm here in the proximal convoluted tubule, the beige part, and I need to reabsorb, say, sodium, I'm reabsorbing it in the peritubular capillary. What if I'm down here? I'm in the nephron loop and I reabsorb water. Where am I putting it? I'm putting it into the vasorecta. Then all of these capillaries, peritubular capillaries, vasorecta, they are all going to go through these vein structures. So again, we're not going to go through all of these veins. What happens to these capillaries? They're all eventually going to drain to the renal vein, and then the renal vein is going to drain to the inferior vena cava. So blood flows through the glomerulus. It forms filtrate. It's going to flow through from the glomerulus to the proximal convoluted tubule, nephron loop, distal convoluted tubule, collecting duct, renal papilla, now called urine. After the renal papilla, we enter the renal sinus. Remember, it's all these structures. So from the renal papilla, we now go minor, major, renal pelvis, ureter, ureter, bladder, urethra. Again, just showing you the pathway. They're starting at the glomerulus here, then they're moving through the proximal Then they're moving through the loop of Henle, then the distal convoluted tubule, collecting duct goes into that renal papilla space, eventually a minor, two minors make a major, two minors make a major, the two majors merge into the renal pelvis, then that becomes the ureter, goes to the bladder, and then eventually exits the urethra. So there are three processes that are happening when we're creating urine. So we are filtering, we are reabsorbing, and we are secreting. So the glomerular filtration step is what we keep talking about happening in the glomerular capillaries. So we're going to see water, dissolved solutes. They're going to enter the capsule space, right? So they're going to go through the glomerular capillaries into the glomerular capsule. And pressure differences is what's pushing the fluid across the membrane. Once it makes it through, once the blood plasma makes it through the glomerular capillaries, it's now called filtrate. Second process we're going to see is tubular reabsorption. So we're going to see items moved, components moved by diffusion or osmosis or active transport. Remember that requires ATP and usually a protein. We're going to see a return to the blood of anything that we reabsorb. So if it's traveling in the PCT and we reabsorb it, we're reabsorbing it into the peritubular or the vasa recta. So the solutes and most of your water gets reabsorbed. Any excess solutes, any waste product, any remaining water will make its way down the tube system. So after PCT, our next section of tube is that nephron loop. So the third process that can happen is tubular secretion. If in the capillaries we have too much of an item, a solute, we can take it and put it into the tube system, PCT or DCT, and that will be excreted. I think this is a really nice image to kind of show you the overview. I think it's easy for us to realize that the proximal and the distal are doing both reabsorption and secretion. We can see the afferent arteriole is moving blood in. The small molecules get filtered through the glomerulus, and now we have filtrate. As this fluid is moving through the proximal convoluted tubule, we can reabsorb, so absorption or secretion. When we get to the nephron loop, this responds to ADH. This is more about water secretion or absorption. So we're pushing water out into the bloodstream, back into the capillaries, or we're pulling water out of the capillaries back into the tube. So remember, if we're pushing something into the tube here, we're ultimately hoping for secretion. If we're pulling that item, if we're reabsorbing it, we're reabsorbing it back into the capillaries because we want to keep it in our blood. So you can see the blood moving in through the afferent arteriole, filtrate happens. The large molecules exit the glomerulus at the efferent arteriole, and you can see that they become the peritubular capillaries, which then becomes the vasa recta capillaries, which then is eventually going to drain via the renal vein, via the inferior vena cava. So proximal convoluted tubule, distal convoluted tubule are doing both absorption and secretion. Loop of Henle or nephron loop here and collecting duct here both have to do with water reabsorption or secretion. So this membrane, they just want to talk about the layers of this filtration membrane. So there is, in the glomerulus, there's a layer of endothelial cells. So these endothelial cells are fenestrated, they have little pores, it allows plasma and dissolved substances to pass. Large items are restricted. There's a good picture coming up. We also have a basement membrane of the glomerulus, and this also restricts the passage of large plasma proteins. We're going to see in just a second that the outermost layer on the glomerular capillary has these podocytes. These are specialized cells. There's thin spaces, filtration slits, and this stops the restriction of most of the small proteins. So the big stuff definitely gets kicked back into the efferent arterioles. Some of the small can move through, but it restricts the majority of them. We also have some specialized cells between the glomerular capillary loops. These can have phagocytic activity to engulf any pathogens. There are some signaling properties, some things that are happening here as well. So I think this is a nice image that you can see the fenestrations, right? So remember, fenestrations are pores. We had talked about sinusoidal capillaries that have those gaping like Swiss cheese holes. Then they want to show you these filtration slits that are occurring, and that this is the cell that's actually coming out and wrapping around this membrane. I think this is an even better image so that you can see, here's the endothelium, and the endothelium here has these pores. So those pores are going to block the really large stuff. So that endothelial, all the large is going to get kicked out. That's a leukocyte, that's a white blood cell, right? Here's an erythrocyte, this is a red blood cell. We can see platelets, we can see large proteins. They will not fit between those endothelial pores. Then we can see there's a basement membrane here. This basement membrane protects from any large proteins from moving through. And then we can see the filtration slits that are created by the podocytes, and that's going to block small proteins from being able to make their way through. So if we're here in the blood, so we're our renal artery coming in, breaking down into afferent arteriole. So I'm going to say afferent arteriole. This is the membrane, this is the glomerular structure. What actually comes out is filtrate. This is the glomerular capsule space. So when I talk about the capsule, it's like this, and then I usually draw the convoluted tuft of capillaries, right? And you have afferent going in, and you have efferent going out. So anything that's dropping into this capsule, we call filtrate, and what could be in there? You can have water, glucose, amino acids, ions. That's when we talked about things like sodium, potassium, calcium, urea, some hormones, vitamins, ketones, which we'll talk about later in digestion, and small amounts of protein. So some items are being shifted through this, through the pores, through the slits, and it creates our filtrate. The big takeaway is what's happening to all of the large items, that they're being ejected into the efferent arteriole, or you could say they're not allowed to pass the filtration membrane, and so they don't have anywhere to go except for the efferent. Remember, these large proteins also help us establish osmotic pressure, right? So this efferent is going to eventually become the capillary, right? And so what causes items to be moved between the proximal convoluted tubule into the capillary? We still have to have this correct pressure difference. So if we dropped off everything, if we pushed everything through the glomerulus, we would not have enough proteins to set up that osmotic pressure difference, and we wouldn't be able to move things in and out of the tubule system that we have. Another factor to think about, this is why would it be bad if somebody said, oh, I have blood in my urine? Why is that bad? So here's your erythrocyte, right? Your erythrocyte, what happens if it's getting through this filtration membrane? There's something wrong with your filtration membrane, right? Imagine having tons of red blood cells stuck in your proximal convoluted tubule, or the nephron loop, or the distal convoluted tubule. It's going to impact the way that the nephron functions, right? So having blood in your urine is not good, and it could be a sign that there's something wrong either individually with certain nephrons, or it could be kidney, overall kidney issue where multiple nephrons are being affected. We produce quite a bit of filtrate. So we're going to have solutes in there, we're going to have water, tiny little bit of protein. The majority is going to be reabsorbed, right? So materials not filtered remain in the blood. They exit the renal corpuscle at the efferent arteriole. So I'm thinking here, these are really large molecules, like your formed elements, white blood cells, red blood cells, platelets. Some of the filtered material is going to get trapped in that basement membrane. Some of it is going to be engulfed, do phagocytosis by those mesangial cells in case there's anything, pathogens in there that we might want to filter out before they get to our functional part of the kidney, which is the nephron. Now we have to talk about some of the pressures associated with this glomerular filtration, and I've kind of alluded to them a little bit, but we have glomerular hydrostatic pressure. Remember, hydrostatic pressure is the pressure of fluid pressing on the walls of the capillary. So the blood pressure in the glomerulus is what pushes the water and the solutes out. It pushes them from the glomerulus into that glomerular capsule space higher than blood pressure of other systemic capillaries. So we're going to see some higher numbers here required for filtration to occur. We also have a large diameter of afferent, so the blood going in is, the arteriole carrying the blood in is larger, and we have a smaller coming out. I also like to think of the fact that this is going to slow down the blood flow, kind of the big picture take home, in order to sort through all of these solutes, right? As the filtrate is moving through the proximal convoluted tubule, we have to have time to recognize for those solutes to bind receptors, for those solutes to be brought from the proximal convoluted tubule into the peritubular capillary. So you can't do this really fast. This needs to be a slow and steady state. The pressure opposing the hydrostatic pressure of the glomerulus, you're going to have the blood osmotic pressure, so those plasma proteins, right, that are creating that osmotic pressure, draws the fluid back into the glomerulus. And then we have the capsule hydrostatic pressure. So this is due to the glomerular capsule. And we're going to see that these forces are overcome by that glomerular hydrostatic pressure. So this is the hydrostatic pressure of the glomerulus. So pushing out, we're at about 60. This is in millimeters of mercury pressure, right? The osmotic pressure from the plasma proteins and the fluid pressing on the wall from this capsule pushing back, so the backward pull, if you will, is at about 50 if you add both of those up together. So you have 60 pushing out and you have 50 pulling back. That gives us a net of plus 10. So what's the overall pressure? The hydrostatic pressure has to be greater pushing out from the glomerulus in order for filtrate to come out of the glomerulus and dump into the proximal convoluted tubule here. We want to have a steady rate at which the filtrate is formed. We call this glomerular filtration rate. This helps the kidneys control urine production. GFR can be influenced by the afferent arteriole if we do vasoconstriction or vasodilation, sometimes altering the surface of the filtration membrane. That's where the little slits were, right? The pores, the fenestrations. They want you to see that there are intrinsic controls inside the kidney and extrinsic controls outside of the kidney. So two ways to control glomerular filtration rate: internal, external. So first, let's look at the intrinsic controls. So intrinsic controls for the kidney to maintain the constant blood pressure and constant glomerular filtration rate, independent of what's happening in the systemic arterial pressure. We're going to look at these two mechanisms. So the first example here of intrinsic control is this myogenic response. So contraction or relaxation of the smooth muscle of the afferent arteriole. So if you have low blood pressure, blood is not moving through the afferent arteriole into the glomerulus enough to create filtrate. So this is affecting your glomerular filtration rate. So decreased blood pressure, decreased glomerular filtration rate. Well, we want that to be more constant. So what can happen? If you have low blood pressure, you're going to see the smooth muscle in the arterioles relax. So you're going to see an opening of the spigot, so to speak. So this is, if the water is just trickling, you go and you turn the handle and make the water go faster. So now the water is, in this case, the blood is coming more forcefully into the glomerular capillaries. So you're opening or dilating, you're increasing the flow of blood, and if you increase the flow of blood, you'll increase the glomerular filtration rate. And now you have this back to normal glomerular filtration rate. If you have increased blood pressure, you're going to do the opposite. So if you have increased blood pressure, the blood is moving so forcefully into the glomerulus, it's going too fast. Your glomerular filtration rate is too high. Again, we want a normal glomerular filtration rate, pretty constant. So if your blood pressure is too high, we're going to limit blood flow. I'm going to do constriction, then the blood is now trickling into the glomerulus. So if we're using the water example, this is the kinking of the hose. If water is going too fast into the glomerulus, I can put a bend or constrict the water and it won't go through the glomerulus as fast. And so then we'll see our glomerular filtration rate drop to a more normal level. In this backup mechanism, this feedback, tubuloglomerular feedback, if glomerular blood pressure, so now I'm in here, if glomerular blood pressure increases, the amount of sodium chloride in the tube also increases. So if I have increased blood pressure inside the glomerulus, I have increased sodium chloride, the macula densa cells are going to detect that, and they're going to send a message, right, to the granular cells, and it can cause vasoconstriction of the afferent arteriole. So increased sodium, because blood pressure is too high, we're going to have constriction, we're going to limit blood flow so that we can get to a normal glomerular filtration rate. There are also extrinsic controls. So as soon as I say sympathetic, hopefully we're thinking brainstem, right? So during exercise or emergency, your sympathetic nervous system is stimulated. It'll result in a decrease in glomerular filtration rate. We're going to see vasoconstriction of afferent and efferent arterioles. We're going to see the JG apparatus, specifically the granular cells, release renin, which we're going to see angiotensin. We're going to see contraction of the Zangial cells. We're going to see decreased surface area of the glomerulus and decreased filtration rate. So the whole point of this, under emergency circumstances or exercise, sympathetic, remember we keep saying decreasing urine production so that you don't have to stop and pee if you're doing your fight or flight. We're also going to conserve water under this stressful environment. So in the last slide with the sympathetic, we saw decreasing. So when sympathetic external controls are initiated, you're going to see a decrease in glomerular filtration rate, a decrease in urine production. In some cases, we might want to increase glomerular filtration rate, and specifically your atrial natriuretic peptide will do that. So this is released from cardiac muscles in response to stretch. We also talked about it in response to high blood pressure. If the pressure is too high, it's pushing on the atria in the heart, and they're going to release ANP. So when this ANP is released, it's saying blood pressure is really high. we need to increase the glomerular filtration rate, push more fluid through. So you're going to see relaxation of afferent arterioles. You're going to see an inhibition of renin. This is good for that high blood pressure, right? So if the blood pressure is too high, we release ANP, limits renin, so then we have a decrease in sodium and a decrease in water absorption. So that would cause our blood pressure to go back down. But we're really focused right now on glomerular filtration rate. So if your blood pressure is too high, we're going to inhibit renin. We're going to have the relaxation of those mesangial cells. We're going to increase the filtration membrane surface, and we're going to increase the glomerular filtration rate. That's going to increase urine volume and decrease that blood volume and blood pressure. So while we're decreasing blood pressure, are we also eliminating more? Right? We're excreting more water. We're trying to make this blood pressure drop lower. So the kidneys are helping increase the glomerular filtration rate to aid in the removal of water so that your blood pressure will go down. I know it's a lot at the end. You'll probably have to go over it a few times. I would definitely make some sort of flow chart for the flow of urine, and for these last hormones, I would kind of divide them up and do more of like an outline. So hopefully that made sense. Ask me if you have any questions, and I'll talk to you guys soon. Bye.